Great. Everybody, let me know if there's issues hearing me. And we'll also be doing a little switch midway through the talk. So Laura Gunzelis, also in the Pollard lab, and additionally in the Kaiser lab at UCSF, has some interesting results to share as well. So with that, I'll get started. The topic of our talk today is about using tools to predict chromatin contact frequency and how we're using that in our lab to predict regulatory consequences of sequence variation in disease, as well as understanding underlying mechanisms and mediators of genome folding. So first, just a review of chromatin contact frequency and its importance in development, especially. I think it's not news to this group that sequence impacts structure within DNA, within our genome, and therefore altering genome sequence can impact DNA structure and therefore gene expression. So for example, we have a sequence here, which presumably has an insulator binding sequence within it, resulting in kind of two distinct structures. And if that sort of insulator binding motif were to be deleted, then perhaps this regulatory separation no longer exists, and a cis regulatory element might make contact with DNA that it usually wouldn't. And this is, in fact, a mechanism of disease. It's been seen before in development with the PITX1 gene, which is restricted to expression in the hind limbs. However, if an important TAD boundary is deleted, then and this TAD boundary binding CTCF and potentially other insulators can lead to, when it's deleted, can lead to the PITX1 gene making ectopic contact with enhancers when it shouldn't, and the outcome is extra forelimb characteristics in the hind limb as a result of this expression change. And so given that we've seen this pattern happening before in development, we're kind of interested in understanding what role 3D genome folding and chromatin conformation could have in other developmental disorders as well. So this is potentially a little difficult to explain, so I guess I won't go too much into it. But we have observed before, Jeff Budenberg in the Pollard group observed that in structural variants that occur in control individuals, so people who don't have any known developmental diagnosis, structural variation tends to kind of avoid highly expressed promoters. So along the x-axis, they're kind of stratifying by expression. So the more important or highly expressed a gene is, the less likely it is that a healthy person would have structural variation at that promoter. And what we see is kind of a broad scale depletion of structural variation around CTCF clusters in these control individuals, which kind of implies, which we already knew, of course, that CTCF clusters are conserved. It's an important element to keep the structural integrity of. And what was found in cases of developmental delay and autism is that structural variation kind of fails to avoid those landmarks, to avoid both promoters and CTCF clusters. So kind of the takeaway here is that structural variation, we do see it happen more in cases compared to controls at these key regulatory features. And so, given all these facts together, we're kind of interested in the implications of 3D genome disruption for developmental disorders. And so kind of, I guess the ideal scenario would be that we could take any patient structural variant and test whether we observe that this, put everything in a stem cell line or a mouse line and observe what gene regulatory changes occur as a result of that variant. But that's obviously prohibitive cost and time-wise. So in response to that problem, Jeff Budenberg and David Kelly from Jeff Budenberg from the Pollard Lab, who is now at USC, and David Kelly from Calico Labs, created the Akita model, which is a convolutional neural net, which predicts high C maps purely from sequence. So given some reference sequence and this Akita model, we're able to predict about a megabase of DNA, how it folds in 3D space. And so we can kind of see first how well is it performing. So this is kind of a comparison of a particular region, what Akita predicted based on reference sequence, and then what we have seen in experimental high C data. For our purpose, we're also interested in testing these mutations in silico, so potentially in disease or other sequences of interest, to see how that sequence alteration leads to a structural change. And so just briefly to kind of establish how we measure this disruption, we kind of use that term as a shorthand for, relatively how different does the high C map look as a result of this in silico variant that we've introduced. And so it's quite a simple mean squared error calculation. And that can also be calculated per bin. And so these bins represent 2048 base pairs, so about 2 KB. And so for each kind of bin in the map, what extent of its chromatin contacts seem to be disrupted or are predicted to be disrupted by the Akita model. And so when we collected all of the structural variants that were observed in CHD cases and kind of scored that disruption level, we also were interested specifically in how those structural variants might be interacting with genes and with the regulation of genes. So we also kind of modified this score based on expression level. Ideally, you might have something like CAGE-seq to show transcription across the whole sequence. And therefore, you can kind of weight your interest in a variant depending on whether a gene promoter in that region has a vastly different contact frequency as a result. And so when we measure this for each of the control structural variants that we had and each of the CHD structural variants we had, what we found was that the CHD cases tended to have structural variants that are highly disruptive. So my axes fell off, but along the x-axis, we're looking kind of at this weighted MSE expression score. So the CHD cases have variants in places that are causing more disruption than the controls. So this again led us to believe that this is possibly an important feature in congenital heart defects. It's not really, not necessarily a common cause, but could potentially explain some cryptic cases. So this is exactly what we used Akita to do. We scored these deletions based on how much they seem to be changing high C maps, and found a few of interest, which I am showing here. And so, excuse me, my camera, here we go. So what we see is often regions of strong insulation making more contact. So the insulation is shown kind of in blue, where contact is being made less often than expected based on linear distance. And sort of these warmer orange colors indicate more contact than would have been expected. And these white stripes are indicating deletions. So we see a few presumed TAD merges, which was similar to the example I showed before. And so all of these kind of had the potential for potentially interesting mechanistic explanations for these patients' congenital heart defects. So from here, we CRISPR engineered those patient deletions into some human iPSC lines, and those have been differentiated into different cardiac cell types. So we have day 2 mesoderm and a few further stages. And this work is being done in the Brno lab with Jingxing Wu and also the Gladstone stem cell core. And so we're kind of in the step where we have created these deletions and differentiated them. And we have some preliminary data on the contact frequency changes, which I will show. And I'm going to focus on a patient deletion that was found in the MESP locus. So MESP is the posterior mesoderm, is the mesoderm posterior genes, very important for heart development. And what we find is that a deletion in this region seems to increase chromatin contacts in the MESP locus and potentially losing contacts with MESP1 and some genes downstream and upstream, as well as gaining contacts within this potentially merged TAD. And so just again, quickly, this MESP1 gene is known to be very important in heart development, and and variants have been shown to cause disease. And also in the region is the KIF7 gene, which misregulation of it can lead to ciliopathies. So we're kind of interested to see how changes in this locus might lead to, how changes in this locus might lead to heart defects, and which genes could be misregulated as a result. So it looks like an image is not loading on this slide. I might try refreshing and I'll be back in just 1 second.
Load a version, Maureen, as well.
Okay. There it is. So what we have in the center here is the capture C data from day 2 mesoderm. So MESP1, as you might imagine, being the mesoderm protein, is most highly expressed in day 2. And there are a few things I wanted to point out, mostly that indeed Akita seems to have kind of correctly predicted the merging of this TAD. So you can see that the region that is in the black box, there's an increase in contacts in the deletion line as a result of the potential kind of TAD merge. And so again, this is quite preliminary data, and we're hoping to dig deeper into it. One reason that we are especially interested in this mesoderm line is because MESP itself has some role in chromatin binding and chromatin shape. So yeah, we have some really exciting questions that we're going to ask with this data, but don't have it yet. So the next slide. And this is where Laura will switch over. So take it away.
Great, thanks, Maureen. So instead of talking about just investigating individual loci, we're now going to jump to how we can use these models to do whole genome unbiased screens and start to uncover sort of families of regulatory elements, and especially transposons and repetitive elements as potential modulators of 3D genome folding. So again, Maureen's spoken about how we can use this model to investigate an individual deletion or an individual insertion, but instead we can sort of tile the entire genome with deletions. In this case, we can delete every 5 kb bin in the entire genome and ask which regions are most disruptive to the 3D genome upon deletion. So if we delete every 5 kb bin genome-wide, we end up with a distribution of deletions where some deletions on the left-hand side don't do anything to perturb genome folding. You can delete this 5 kb region with no difference between the predicted reference map and the predicted map with the 5 kb deletion. On the other side, some 5 kb bins produce large, widespread changes to 3D genome folding, like massive structural rearrangements. And for the most part, the ones in the center produce small changes or no changes, like focal changes or perhaps the loss of a stripe or a loop. But we're really interested in knowing what is causing these most disruptive deletions to the 3D genome in sort of this unbiased fashion. And we can tile these deletions across the genome to have a disruption track where the disruptability or stability of the 3D genome is not uniform. So we have some regions that are more sensitive up here and some regions which are more robust to perturbation down here. And we can layer on, say, gene bodies and CTCF, and we notice that regions that lack CTCF entirely don't seem to correspond to valleys in this perturbation track, meaning that if it lacks CTCF, deleting the region is unlikely to have any strong effect. It's very robust to disruption. And we also noted that this disruption track is very well correlated to sort of a completely orthogonal measure, A and B compartment. So A compartments seem really sensitive to 3D genome disruption, and B compartments seem really robust. So we can make mutations and alterations within B compartments, and it doesn't seem to have a large impact on 3D genome compared to A compartments. Now we can ask, where are these most disruptive disruptions falling across the genome? So we can compare sort of the top 1% to the bottom 1%, most and least disruptive deletions, and ask where they fall within ENCODE candidate cis-regulatory elements. And very expectedly, we find that a majority, over 50%, of the most disruptive deletions fall within 5 kb of a CTCF-bound distal enhancer, which makes a lot of sense. But sort of surprisingly, we also noted that a large percentage, over 20%, don't fall within any ENCODE cis-regulatory region at all, leading us to wonder what other mechanisms could be potentially driving these extremely disruptive 3D genome deletions if they're not a known ENCODE regulatory region or CTCF-bound region. We also noted that repetitive elements, which make up over half of the genome, also seem to correspond to peaks in this disruption track, similar to compartment and CTCF. So we noticed that, say, bulk clusters of ALU elements, like seen here and here, correspond to peaks in our disruption track. And indeed, we noticed that if a region contains no ALU element whatsoever, we sort of have a baseline level of disruption. And if we add an ALU element, so if we look for regions of the genome that contain only one ALU element, we find that these regions are more sensitive to 3D genome mutation. And if we keep adding elements, we see this trend where the more ALU elements we have within a 5 kb region, the more sensitive this region is to disruption. So this trend looks very similar to what we observe with CTCF, where the more CTCF elements we have, they seem to work in tandem to provide sort of the scaffold for 3D genome architecture. And we note that this isn't only the case with ALU elements. We also note this with a variety of repetitive elements, as well as small non-encoding RNAs and tRNAs, where the more elements you have, the more disruptive the region is upon deletion. But it's not true for all repetitive elements. For example, the L1 line family, there seems to be no trend or even potentially a slightly negative correlation with the number of elements and how disruptive the region is to 3D genome folding. So instead of deleting 5 KB bins, we can now individually target repetitive elements. And to do this, we delete over 1 million elements in the repeat master database by individually deleting them and comparing a wild type to a reference map. And the result is a wide distribution of how disruptive repetitive element families are to 3D genome folding. But we note that a substantial amount of them are greater than you would expect from size-matched random deletions, potentially supporting their role in 3D genome folding. And we can ask, are these potentially mediated by CTCF? We know that CTCF is already located within lots of repetitive elements. So we would potentially want to note this as a mechanism. So for example, upon deletion, we have the loss of boundaries, like in this hat tip 100 element, where we have the loss of this boundary. We have loss of boundaries again, but also loops, as seen here in this ERV1 deletion, as well as gain in contact, as seen like downstream here in this allo deletion. But surprisingly, when we look at the top 10% of these most disruptive deletions in this 1 million element screen, we see that only a minority of them contain CTCF. And in fact, the vast majority contain no elements compared to one or more CTCF elements, really leading us to wonder whether these might be mediating 3D genome folding through some other route, or potentially in conjunction with downstream or upstream CTCF. And to demonstrate the power of how we can use these types of models to investigate mechanisms, we can not only delete elements, but we can also insert them back into the genome. So here we have one example of a particularly detrimental MER91B deletion. So here's the reference map on the left with no deletion whatsoever. And when we delete this element, which is around 250 base pairs, we see the complete loss of this boundary here. And here's the difference between the two maps, where we have a striking loss of a boundary centered at the location of deletion. If we insert a sequence, or just a random sequence that's the same length, we don't have any gain of the same phenotype. We have the same loss of contact as we observed in the original deletion. We can insert CTCF instead. And again, we don't see that CTCF is able to rescue back the phenotype. But we can instead insert back a very similar MER91B sequence that's also conserved. And we notice that this MER91 sequence is sufficient to rescue back the original contact map, even though the element itself is different. So demonstrating that the model is internally consistent, and we're able to use this model as an experimentalist would for investigation and discovery. So in summary, Maureen and I talked today about how we can use Akita and these deep learning models for really powerful biological investigation. And so we can do this in the case of known variation, as Maureen spoke about, or we can do this in a completely unbiased manner for discovery, like I spoke about. And so this is really pointed to potential mechanisms in heart disease, and also pointed to repetitive elements as potential mediators of 3D genome folding. And I think with that, we can take questions.